Biochemistry and overview. Biochemistry is the study of the chemical substances found in living organisms and the chemical interactions of these substances with each organism and the chemical interactions of these substances with each other. It deals with the structure and function of cellular components such as proteins, carbohydrates, lipids, nucleic acids, and other biomolecules. There are two types of biochemical substances: bioinorganic substances and inorganic substances. An example of bioinorganic substances are carbohydrates, lipids, proteins, and nucleic acids, while inorganic substances are water and inorganic salts. Complex bioorganic or inorganic molecules, enzymes, vitamins, DNA, RNA, and hemoglobin, and etc. As isolated compounds, bioinorganic or bioorganic or complex substances have no life in and out of themselves. Yet, when these substances are gathered together in a cell, their chemical interactions are able to sustain life. Plant materials. It is estimated that more than half of all organic carbon atoms are found in the carbohydrate materials of plants. Human uses for carbohydrates of the plant kingdom extend beyond food. Carbohydrates in the form of cotton and linen are used as clothing. Carbohydrates in the form of wood are used for shelter and heating in making paper. Occurrence and functions of carbohydrates. Different objects such as sheets of paper, insect skeletons, fruits, cotton fabrics, and ropes have one common feature: they all contain carbohydrates. The chemical structure of carbohydrates, with their many hydroxyl groups and the ability to assume various spatial configurations, make it possible for them to form nearly unlimited combinations with other carbohydrate molecules, as well as with proteins and lipids. The resulting structures perform important biological functions. Almost 75% of dry plant material is produced by photosynthesis. Most of the matter in plants except water are carbohydrate material. Examples of carbohydrates are cellulose, which are structural component of plants; starch, the energy reservoir in plants; and glycogen, or animal starch, found in animal tissues and human body in smaller quantities. Plant products are the major source of carbohydrates, and average human diet contains 2/3 of carbohydrates. Recommended percents in the daily diet. Recommended carbohydrates is 60%. Recommended sucrose less than 10%. Usefulness of carbohydrates is their ability to produce energy when they undergo oxidation during respiration. Storage carbohydrate in the form of glycogen provides a short-term energy reserve for bodily functions. Carbohydrates supply carbon atoms for the synthesis of other biochemical substances, or proteins, lipids, and nucleic acids. Carbohydrates also form a part of the structural framework of DNA and RNA molecules. Carbohydrates link to lipids, as discussed are structural components of cell membranes. Carbohydrates linked to proteins function in a variety of cell-to-cell and cell molecule recognition processes are useful markers for antibodies. What are carbohydrates? It is a group of organic compounds occurring in living tissues and foods in the form of starch, cellulose, and sugars. The ratio of oxygen and hydrogen in carbohydrates is the same as in water. It typically breaks down in the animal body to release energy. What is the general formula of carbohydrates? The general formula for carbohydrates is CxH2OY. Definition of carbohydrates in chemistry. Chemically, carbohydrates are defined as optically active polyhydroxyaldehydes or ketones, or the compounds which produce units of such type on hydrolysis. Carbohydrates are also called saccharides, which is in a Greek word and it means sugar because almost all the carbohydrates have a sweet taste. Sources of carbohydrates. We know carbohydrates are an important part of any human's diet. Some common sources of carbohydrates are potatoes, maize, milk, popcorn, bread. Here are a few examples of where you'll find the most carbs. Dairy products like yogurt, milk, ice cream. Fruits, fruit juice or whole fruit. Grains like cereals, bread, wheat, and rice. Starchy vegetables like corn and potatoes. The main classification of carbohydrate is done on the basis of hydrolysis. This classification is as follows. Monosaccharides. These are the simplest form of carbohydrates that cannot be hydrolyzed any further. They have the general formula of CH2ON. They consist of one sugar containing 3, 4, 5, 6, and 7 carbon atoms and are usually colorless, water-soluble, crystalline solids. Some monosaccharides have a sweet taste. Examples of monosaccharides include glucose or dextrose, fructose or livulose, galactose, cellulose, and ribose. In disaccharides, a further classification of oligosaccharides. These give 2 units of the same or different monosaccharides on hydrolysis. A sugar or a carbohydrate composed of 2 monosaccharides, oligosaccharide. An oligosaccharide is a saccharide polymer containing a small number or typically 3 to 10 monosaccharides. Example, sucrose on hydrolysis gives 1 molecule of glucose and fructose each, whereas maltose on hydrolysis gives 2 molecules of only glucose. In polysaccharides, the final category of carbohydrates, this gives a large number of monosaccharides when they undergo hydrolysis. These carbohydrates are not sweet in taste and are also known as non-sugars. They are insoluble in water and have no sweet taste. Some examples are starch and glycogen, etc. Chirality, handedness in molecules. Chirality is a geometric property of some molecules and ions. A chiral molecule or ion is non-superposable on its mirror image. The mirror images of a chiral molecule or ion are called enantiomers or optical isomers. Individual enantiomers are often designated as either right-handed or left-handed. Most monosaccharides exist in two forms, a left-handed and a right-handed form, same as two hands. Two types of objects, superimpossible on their mirror images, are images that coincide at all points when the images are laid upon each other. A dinner plate with no design features, or a chiral. Non-superimpossible on their mirror images is called chiral or handedness. For example, tartaric acid contains two asymmetric centers, but two of the isomers are equivalent and together are called a meso compound. This configuration is not optically active, while the remaining two isomers are D and L mirror images, enantiomers. The meso form is a diastereomer of the other forms. The four groups attached to the atom at the chiral center assume a tetrahedral geometry, and it is governed by the following conventions. Vertical lines from the chiral center represent bonds to groups directed into the printed page. Horizontal lines from the chiral center represent bonds to groups directed out of the printed page. In a Fisher projection formula, a chiral center or carbon is represented as the intersection of vertical and horizontal lines. Functional groups of high priority will be written at top D and L system used to designate the handedness of glyceraldehyde enantiomers. Stereoisomerism, enantiomers, and diastereomers. Stereoisomerism is the arrangement of atoms in molecules whose connectivity remains the same, but their arrangement in space is different in each isomer. The two main types of stereoisomerism are diastereomerism, or including cis-trans isomerism, optical isomerism, also known as enantiomerism, and chirality. In enantiomers, they are mirror images of each other and are non-superimposable. They always have a different RS configuration. They have one or more stereocenters. They have the same chemical and physical properties. They all possess optical activity, although they rotate light in opposite directions. Diastereomers, they are not mirror images of each other and are not non-superimposable. They have the same RS configuration, at least on one stereocenter. They have at least two stereocenters. They have different chemical and physical properties. Not all diastereomers possess optical activity. Designating handedness using Fisher projection formula. Fisher projection formulas, a method for giving molecular chirality specifications in 2 dimensions. A Fisher projection formula is a two-dimensional structural notation for showing the spatial arrangement of groups about chiral centers in molecules.